Hi, I'm Simon Prochnick, and I work for the Plant Genome Program at the Joint Genome Institute. Volvox is one of the most beautiful algae that I've ever seen. It's a wonderful little dust speck-sized green swimming alga. It's not going to overtake an Olympic swimmer, but for a plant swimming around in a pond, it's quite fast moving. It swims with its fantastic rolling motion. So as the sort of round Volvox organisms swim, they have this characteristic kind of corkscrew rolling motion. And the name Volvox actually comes from the Greek fierce roller. I'm not quite sure why it's fierce, but they certainly do roll. This is actually a really nice project because the Chlamydomonas genome itself, which was published in 2007, revealed some very interesting things about the algae. It revealed that they share genes with animals. It reveals that they share genes with bacteria. And obviously, as they're plants, they share gene with plants. With Volvox in hand as well, what we're able to do is compare these two green algae, and now we can do interesting things like look at what genes are shared between the two algae and what genes are unique in the two algae, and what genes are just in Volvox and what genes are just in Chlamydomonas.
When you look at the genome sequence, all you see are a sequence of A's, C's, G's, and T's, and it means nothing until you ask specific questions with specific information. So the work that I've been interested in all my life which is understanding the origin of multicellularity in this group, has just begun. But it's now going to be, the answers are going to be much more readily accessible.
The Volvox genome is slightly bigger than the Chlamydomonas genome, and that's mostly because repeats are expanded. But apart from that, as my boss, Dan Roxar, likes to ask me, and he kind of teases me with this, he says, if I put the Chlamydomonas genome under one cup and the Volvox genome under another cup, and I mixed them up and then took the two cups away, could you tell which is which? And the answer is no, I couldn't. And I spent 3 years trying to find a difference. And at the end of 3 years, the results that I and a team of nearly a dozen collaborators found is that there's actually very little difference at the level of protein coding genes.
Our project with JGI to sequence a mating locus of Chlamydomonas and Volvox turned up a rather remarkable difference between them. So overall the genomes are very similar, but Volvox has really expanded its sex-determining locus and turned it into a sex chromosome, and that's in turn had all sorts of downstream effects on evolution of the genes that are in it. And this was a project that was enabled by one of these community sequencing proposals that JGI did, and it meshes really nicely with the genome sequence. So I think the two projects were highly complementary to each other.
One class of proteins really stood out as not showing quite the same distribution. And these are proteins that can be grouped into families. And these are protein families that are only found in the Volvocene algae. So they're found in Volvox and Chlamydomonas and their cousins, but not in other plants, and they're not found in animals, they're not found in fungi. And when we stared hard at these protein families, we found that on average they had more members in Volvox than they did in Chlamydomonas. So it looks like these special protein families that are Volvocene algae specific have expanded in Volvox and have not expanded in Chlamydomonas, or potentially have lost members in Chlamydomonas. And this kind of evolutionary signal is the kind of signal you see if the proteins are useful for something that Volvox does that Chlamydomonas doesn't. And so this is a kind of little hint that somewhere in these, I think there were 1,835 families, somewhere in these families is maybe going to be some of the clues as to exactly why Volvox is multicellular and why Chlamydomonas is not.